CRISPR was the 2015 Science Breakthrough of the Year. Everybody in the scientific community is abuzz with the new possibilities created by this process. CRISPR is the first ever system that will allow us to edit genomes. Of course, this is a really complicated process that many have yet to understand, but I'm going to break it down into smaller words and explain the basics. CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's a lot to say over and over again, hence the term CRISPR. The CRISPR, or Cas9 system, is based off of a process used by bacteria. The bacteria land on your cell like a little spaceship. The spaceship then sends into your cell a protein called Cas9, and inside that Cas9 protein is a strand of RNA.
To understand RNA, we must first understand DNA. DNA, or deoxyribonucleic acid, is a molecule that carries the genetic instructions in all known living organisms. DNA is assembled in a shape called a double helix, which looks something like this. If we were to take this double helix and break it down into smaller parts, we would find something called nucleotides. And if we took those nucleotides and break them down into even smaller parts, we would find something called nucleobases. There are 4 different nucleobases: thymine, adenine, guanine, and cytosine. The nucleobases on one side of the double helix match up and pair with the nucleobases on the other side. But there are some rules to this bonding process. Guanine can only bond with cytosine, while adenine can only bond with thymine. Because of this, if we only have one strand of the double helix, we can then synthesize the other strand. This leads us to RNA. RNA is ribonucleic acid, and much like DNA, is a molecule that carries genetic instructions, only there are a few key differences. The first is that RNA is single-stranded. The second is that in RNA, the nucleobase thymine is replaced with uracil. And lastly, the DNA is kept inside the nucleus at all times, while RNA is allowed to travel throughout the cell. This makes RNA a messenger of genetic information. Therefore, if we should need to go into a cell and search for a specific DNA sequence, we can then send in a strand of RNA with the mirrored code. The Cas9 protein, once inside your cell, attaches itself to your DNA. Cas9 then unzips your DNA so that the RNA has a chance to search for the mirrored DNA sequence and zips up the double helix on the way out again. Once the correct segment has been found, Cas9 then makes a double-stranded cut on your DNA and detaches from the target segment. Think of the RNA as the president, Cas9 as his bodyguards, DNA as a line of people waiting to meet the president, and the target sequence as a spy in that line of people that has crucial information. The president is waiting to hear a password from this spy. The bodyguards separate the crowd of people into 2 lines that break around the president and reform again as they exit. Once the president hears the password from that spy, he signals the bodyguards, and the bodyguards cut off the remaining line of people, separating them from the rest of the line. Once Cas9 has made the cut to your DNA, your body tries to repair the damage. Only most of the time, this repair includes mutations. The great thing about CRISPR is that it can improve this repair by adding in a specifically programmed sequence, and this repair can be even better than the original. That's the crazy part, is that the CRISPR repair can be much better than the original. The CRISPR cut-and-paste genome editing method could eradicate genetic syndromes such as cystic fibrosis, sickle cell anemia, and HIV. We might even be able to extrapolate human abilities so that we can explore the depths of the ocean without the need of scuba gear. We don't really know the extent to which we can go with CRISPR yet, but what we do know is it'll change everything that we have known thus far.